I'm Bill Hammond. I'm at the University of Florida, where I run the ecophysiology lab there. And I titled the seminar today, "What Kills Trees?" You know, this really is the question that has kept me up now since I started pursuing it early in the days of my PhD. Today, I want to talk about how we determine some of the climate drivers of tree mortality, what some of the important mechanisms of tree mortality are. But I want to acknowledge that this is like a complex problem. And what kills trees is actually a really difficult thing to pin down. Besides what I'm going to focus on today, heat stress and drought stress, there are, of course, pathogens and pests which get intensely and scrutinized and studied here at FABI at the University of Pretoria. Besides this, other disturbances like fires and floods, deforestation, there are compound threats, there are manifold threats to forest survival. But in the Anthropocene, two things that seem to be doing a lot of the driving from an abiotic sense are intensifying heat and drought stress. And so that's primarily what I'm going to talk about. Now, when I talk about tree mortality, I don't mean the background rate, how many trees die a year. I'm talking about elevated levels of pulsed mortality. So the mass mortality of individual trees in an area or a population. Here's some examples from a recent kind of global synthesis that I did on the topic, showing this is dead sugar pine in Sequoia National Park. This is loblolly pine, Pinus taeda, in plantations in Corrientes, Argentina. This is from just a couple of years ago, a paper out last year from Gabriel Gatica and his group there in Argentina. And even places that are typically wet can get hot and dry enough that we can see pulses of forest mortality like this. And so what I set out to do several years ago was to gather all of the peer-reviewed published observations of tree mortality events like this across planet Earth. And that's what I'm playing this animation for you now. The decades are colored by year. And in total, by the time we get into, I think 2018 or so, when this will stop, what you'll see is forest mortality events like this are happening across every forested continent of the Earth. There's over 157 species that are dominant at these sites that have pulses of mortality like this, that covers more than 40 genera of trees as well. This happens to angiosperms and gymnosperms alike. The animation hung up for some reason, but you get the point. It looks like the planet has a case of the measles or something. It's not good. And so this is kind of a globally ubiquitous phenomenon. If we take that same data, and a big part of the study was to kind of precisely georeference where on Earth these pulses of mortality were happening and also identify when in time the trees died, we can now take those coordinates and put them instead of geographic space into climate space. So here's annual precipitation from 0 to 4 meters on the Y, and annual temperature from -10 to +30 C. Trees don't grow in the tundra, but in every one of these really coarse biomes that Robert Whitaker put forth in the 1970s, we can see that these mortality events have happened. So the take-home message here is basically whatever the average climate is of a forest, eventually pulses of elevated temperature and intense drought stress can lead to these kind of mortality events. This is work that we shared not only in journal publications, but with the general public in National Geographic a couple years ago when the data came out. And so this is something that's really a global phenomenon. But as a physiologist, it's not enough for me to know that the situation's bad. I want to put a number to it. I want to know how dry is too dry and how hot is too hot. How can we quantify this? So by synthesizing these global data, we were able to kind of ask this question by investigating the long-term climate at each of these locations and the climate during the years of tree mortality events. So we took this map, which has almost 5 decades of observed tree mortality events on Earth, and aligned it all to a single common year of tree mortality. And that's one of the first results I'm going to show you here. It might be a little small on the screen, especially if you're in the back, but this y-axis is 6 different climate metrics. They've been standardized as a z-score, so that the units on the x here are standard deviations. The top 3 are the monthly maximum temperature, the vapor pressure deficit, the climatic water deficit. The bottom 3 are precipitation, soil moisture, and the Palmer Drought Severity Index. It's kind of an analog to SPEI, if you know that. It's a more long-term integrated drought stress index. Anyways, these are 6 different climate metrics that we usually think of as being associated with drought or heat stress. When the data points I show you are to the left of the center line, that means they're significantly wetter or cooler than long-term climate trend during that year. When they're to the right of the line, they'll be colored red. That means they're significantly warmer or drier for that relative metric. And this is showing you the 4, 3, and 2 years prior to the mortality of Earth's forests. And so now I'll reveal the middle panel here, which is going to show you the years bounding the year that the mortality was observed across Earth's forest in the last 48 years. Here what we see is this kind of strong signal begins a year before the mortality event and persists even a year after. This is cohesive across all 6 of those climate metrics. And this basically represents what we call a hotter drought fingerprint, or a quantification of how much hotter and how much drier in this kind of multi-dimensional, multi-metric way is it when we see these pulses of elevated forest mortality. The years after that, things kind of return to normal, where we don't have that strong signal agreement among these multiple climate metrics. In the supplement to the paper, you can look, like 20 years into the past, and you never see a fingerprint like this show up along that long-term climate record. So we do feel like this is a pretty cohesive and consistent signal. We also provide versions of this in the paper in Nature Communications that breaks it down by biome if you're interested. We also took those global forest mortality observations and looked at a couple of climate scenarios that are different than the observed climate. So the observed climate at these sites is in blue here. These density distributions for soil drought stress through that Palmer drought index, atmospheric drought through the vapor pressure deficit anomaly, and the direct effects of temperature stress through the maximum temperature anomaly. We retraced the historical climate by warming it by, on average, either +2 or +4C, using like an ensemble of 27 climate models. And what we found was it's not just that, that average warming of the climate causes the mean to shift over. It really pushes the tails of these distributions out. And so what we're worried about with future climate is intensified extremes, right? Not just that it might get hotter on average, but that the single hottest day might be much, much hotter, maybe 10 or 15C warmer than the hottest day today. And so this is the kind of thing that can lead to projections here where we say, okay, our observed climate, which is about 0.7C at the time of 2 years ago, this paper being published about pre-industrial, we expected a mortality event at one of these sites on the Earth about 1.5 years per decade. If the Earth warms to +4C, we might expect that 4 out of every 10 years would have a mortality event like this. So the increase when the planet warms to +2C is going to be moderate, about a 22% increase in pulses of the climate that has been historically causing tree mortality. But by +4C, it goes up 140%. So this really irresponsible line that I fit to 3 data points, students in the room, don't ever do that. I only did it to draw your eye to the fact that this isn't a straight line, right? It's a curve. And we all learned something about curves and how it would be best to avoid them when it deals with mortality during the COVID pandemic, right? Every effort we can make to further limit the warming of the planet is probably going to give us exponential benefits to forest survival. Just want to call that out. And so we know the planet's getting warmer on average. You've probably seen something like this. This is the change in global temperature since 1850 when the steam engine was invented, which is largely marks the beginning of the Industrial Revolution forward to today. The last couple decades have been especially intense in the amount of warming that's happened over pre-industrial. But it's not just that it's getting warmer on average. These hottest days are getting hotter. And so these are maps from the latest IPCC report of the annual hottest day temperature change. And by the end of this talk, I'm going to show you some maps for what the future looks like for the hottest single day in South Africa. I made those maps last night because I'm the kind of guy that finishes something when it's due. And so as the rain comes on average 2, 3, or 4C, you're going to see that the annual hottest day might go up as much as by 7 degrees C in many places, including places like where we are today. And so this is a cause for concern. The sort of precipitation is a little more variable. Some places are going to get wetter, like in the Congo. That's in contrast to tropical forests in Brazil, for example, that are going to be expected to continue to get drier. And so different parts of the world are going to experience drying along with this intense warming. In all places, pretty much the variation in when the precipitation falls within a year is definitely going to be affected by all this changes in energy in the system. And so we kind of are able to quantify how hot is too hot and how dry is too dry, but a problem that remains is how can we identify trees that would tolerate or maybe be able to be more resilient to hotter drought stress that's coming now in the future, that we've seen these projections. And this gets into, I think, why Bernard asked me to come today was to share with you some of the methods I use as an ecophysiologist to screen or phenotype through different plant materials and understand what might be more or less resilient to these stresses. And so the solution that I'll put forward for you today is first, I'm just going to walk you through a bunch of those different methods. It's going to be a little bit like a buffet. And so if you don't see, if the slide I'm showing you don't like, just wait, maybe you'll like the next one or something further down the line. And you know, what we can measure. But then finally, I'll put that all together for you and show you a case study and a recent funded project I have through the USDA looking at mapping climate resilient growth and survival traits in loblolly, which is the most planted tree in North America. Yeah, and so when I think about what traits we need to be measuring, I want to share with you a simplified framework from a paper I put out at this point a long time ago. And on the y-axis here is mortality risk. So if you're at 0, this little dashed line means there's not a chance you're going to die, right? Whereas 100 means like you're cooked. This is not good, it's game over, right? And I'd really like to know how this mortality risk evolves over some duration of stress, right? So this is just a conceptual figure. And when we think about especially drought and hotter drought stress, we're often thinking about what's happening with water. And so I'm breaking this into 2 phases. The first one here, the growth limitation phase, I'll call it for simplification's sake. Here, water loss through the plant is mainly still happening through the stomata, and so plants are able to kind of actively regulate during this growth limitation phase their water use by adjusting their stomatal aperture. And traits that are going to influence how long we're in the growth limitation phase and not the next phase that I'm going to show you in a moment are things like knowing what's the point at which the foliage of this plant is going to lose turgor. What's the turgor loss point or the wilting point of the plant? Also knowing things like the optimum temperature for carbon assimilation and stomatal conductance and water use efficiency. Plants like you and I and all life on Earth are thermally optimized. So knowing how these things interact, not just with water, but with temperature is really important too. Eventually, though, once we get out of this phase and the stomata are closed and the leaves are wilted, we cross this like incipient mortality risk line that I've drawn here, and we enter into this mortality risk phase. Now, once we cross this threshold, your chance of dying is no longer 0, right? And it may not immediately go up very fast. I don't know, that's a bad spot to be. Okay, I won't stand there. And I've drawn here on this conceptual framework just 2 potential lines. One that's a red dashed line, which represents maybe a heat wave atop a drought, and a blue dashed line that represents maybe a drought without a heat wave. And so how rapidly after that incipient mortality risk point, you reach this point of no return that I call it, which is when it's more likely that you'll die than that you'll survive, might depend on things like how hot is it? The traits that we can measure to understand the shape of these curves and make predictions for a species are going to be things like how vulnerable is your xylem? How much water deficit can your vascular system withstand before the continuous threads of water are broken, either by air entry into the xylem or cavitation, spontaneous phase change. Knowing the water loss rate, here instead of stomatal conductance, stomata are done with their job in the mortality risk phase, really. But like you and me, stomata aren't perfect. And so sometimes they leak. And those leaks, along with water that's leaving across the cuticle, or maybe the water that's leaving across the bud, can add up to this what we call residual conductance. And if we know this residual conductance rate, and we also know the capacitance or the stored water pool that's available in the plant or the organs we care about, we can start to really describe this shape pretty well. Of course, all of that's going to really depend on a surface area basis of the plant. So we have to work really hard to get good, accurate measurements of leaf area or canopy area. And so that's kind of the framework for thinking about some of the things we need to measure. But when it comes to climate-induced tree mortality, it often has been called drought-induced mortality. And in fact, before the paper we published the hotter drought fingerprint 2 years ago, we didn't really have a quantitative evidence for the role of heat in these global mortality events. And so something that we're really getting into measuring a lot more these days are thermal limits. And here I'm showing you one way that this can be done. Basically, in principle, what you need to do is take leaf material from a plant or really any organ, depending on how you quantify vitality, expose it to a range of temperatures. Here it's going from 30 to 60 degrees Celsius at 10 different temperature stops along the way. And then you probe the vitality of that sample afterwards. We do that by We have these little 3D printed leaf cassettes. We put 3 replicates at each of 10 temperatures. So this might be 30 degrees, 35, 40, and so on until we get to 60 C. And then we take a chlorophyll fluorescence image. You could also use just a regular point source fluorometer for this measurement. It would just take 30 times as long. So the high throughput part here is just by turning this into an imaging platform, we can use custom written R code to extract all the values, fit a model, and extract the traits of what is the critical temperature, or the T50, the temperature beyond which 50% of that vital signal is lost. Being able to do this in a high throughput way means instead of measuring traits like this at the level of like species, now we can start to screen a lot of materials within a species. So for example, in my lab right now, we've sampled more than 50 different species of woody trees. We found a range of variation in the T50, the kind of killing temperature, between 42.5 and 62.5 degrees C. The thermal tolerance champion we found in the Brazilian Cerrado during a research visit last year. Pinus taeda, loblolly pine, we've measured over 50 provenances of that and found a 6 degrees C variation in the thermal tolerance just within a single species. So this means this is probably something that when we look at improving, silvicultural lines, this is not a kind of trait that we're making selections on in breeding programs when we go after yield. And understanding how this varies or in the materials that are planted out on the landscape is really important, especially in long-lived woody plants. Eucalypt might get harvested in 6, 7, or 8 years, but a pine, it might be 2 decades from now. 2 decades from now, I don't want to make everyone feel old, but that's like 2045. Right? And we do the climate I'll show you climate projections for 2045 by the end of this. We also need to measure that residual conductance. We build in the lab these climate time machines. These are modeled after a technology called the drought box. And basically, these are boxes where we can insulate plant samples and weigh them. We have little weighing lysimeters where we can measure the plant over time, so we can see sample mass decline as environment changes. We can very precisely control temperature across a range that like a growth chamber can't. Most of those max out at 40, 45 C. We can take these boxes from 20 to about 73 C if we want. And importantly, you can keep the humidity constant, or you can alter it up or down independently of temperature. So you can keep vapor pressure deficit constant or alter it or control on that condition too. These are kind of modular, stackable design versions of these things. Another thing that's really nice about these is we can put whole plants in them. These posts on the bottom also have load cells that hold much higher mass. And so we can put whole intact, seedling and small sapling plants in and see how they respond to a heat wave, for example. So we call these a time machine because we can put the plants in, we can set the climate environment to the year, 2100 under whatever climate scenario we want, and then see how they perform under those conditions. It's really interesting because now you can take plants like these pines, put them through just a 6-hour heat wave, experiencing just the 6 hottest hours of a day, and you can kill them in dry air, even if their soil is wet. I'm going to show you a lot more data about that at the end of this talk. Another thing we need to do when it comes to physiological phenomics is we got to find a way to speed it up. If you've ever, raise your hand if you've ever measured water potential on a plant. I don't know if that's, there's a couple people, right? It takes a little bit of time. Right. This trait right here, the turgor loss point, we often quantify doing what's called a pressure volume curve. And so you measure the water potential and then you weigh the leaf, and you measure the water potential and you weigh the leaf. And eventually you have to wait a couple hours in between measurements because desiccation is taking longer as the stomata are closed and you're on that residual conductance. And so getting a nice curve like this might take 3 or 4 hours. In a species that dries quickly, in a conifer, it might take 3 or 4 days. And so this is not very high throughput. But this is a really critical trait, like I said earlier, for knowing when are plants going to stop growing and start dying. And so to quantify that, we've been working on kind of leveraging the fact that as plants are drying out, there are changes in their anatomical structure as cell volumes shrink and swell and as water contents decrease, and trying to understand, can we build spectral models to predict this same thing? And Dr. Castillo, one of the postdocs in my lab, has built independent models for predicting relative water content and water potential that use different wavelengths. And that now we can basically dry out samples and estimate the turgor loss point without ever having to pick up the pressure chamber once we've calibrated a model well at the species level. We published this last year in New Phytologist, so you can check that out. We call it spectral ecophysiology. And it's one of the steps that we think will be necessary to put us to the point where we might be able someday to screen hundreds of lines, for example, or hundreds of families for important traits. We started with the turgor loss point. There's a suite of about 14 physiological traits we're presently trying to build models like this for. To know the drought vulnerability, or when does the xylem stop functioning, we use two instruments in my lab. One is a Cavitron that we built. This is an instrument that's been around maybe since the early to mid 1990s, where we take woody branches off of plants that have xylem conduits that are shorter, that are short enough that they're not open on the ends in a 28-centimeter long sample. And in this rotor, we spin those branches at such a high speed that the centrifugal force causes an exact xylem tension in the middle of the sample. So instead of setting a branch on the counter and waiting for it to dry down to a certain amount of xylem tension, like I did in my PhD, it was very painful and also boring. Now we just put them in the machine, like a washing machine, and just like spin them up to the tension we want. We use a high-speed camera to measure flow through the sample while it's spinning. Not compatible with all anatomies, but in things like conifers, it works quite well. To quantify this trait of when does embolism first appear and how is it spread in space and time, and eventually run to completion, we use these cavitation cameras, or CAVI cams. These were designed by folks at the University of Tasmania in the Broadrib lab. They have open source plans, so we 3D print our own. We have about 40 of these in the lab. We use them both for vulnerability monitoring and also as optical dendrometers. I'll show you some data on that in a second. Another thing that we do in the lab is some of this micro CT work. So sometimes you don't want to cut a plant to be able to see what's going on with its xylem function, because the hydraulic system is under a negative pressure. It's equivalent to a tension, and when you cut it, you significantly disrupt that system. And so using these kind of X-ray systems, we can look inside of a plant without having to cut it. And because of the differences in density of air and water, we can see which conduits are filled with air and which are filled with water. This is from a eucalyptus, which I thought might be topical here in South Africa. I did this experiment when I was in Israel in my PhD. All the xylem here closest to the bark in the vascular cambium is a little bit brighter because I fed the tree via its roots some stain solution, iohexol, that brightens in X-ray. And basically, after an extreme drought that killed most of the canopy, the tree recovered, but not by refilling any of its conduits, but by radial growth. It grew new pipes. And so we know that one of the keys to trees being able to withstand extreme drought stress and recover is that their meristem, their vascular cambium, needs to maintain a water supply. Those cells need to live through the stress event, one, and two, there needs to be a functional water supply, or at least enough water supply that cambium can divide. Xylogenesis can lead to maturation of new conduits, and thus kind of these legacies of dysfunction can eventually be erased by regrowth. We also just established last year this plant array at the University of Florida. It's a physiological phenotyping platform where every plant sits atop a weighing lysimeter. There is a watering system where we can water each pot gram by gram. And inside of each pot sits a sensor for soil moisture, soil temperature, and bulk soil electroconductivity. Then on top of the plants, we'll install, these cavitation cameras on leaves, or maybe a psychrometer on the stem, so we can track other physiological variables we want to measure. This system's showing a lot of promise. We're still working it into our experimental flow, but I thought I'd give you an idea of what some of the data looks like recently. This is from about a 2-meter tall magnolia sapling. We've got 100 pots in our small pot array, and in the room behind, there's 32 pots that are 25 liters each. So we can go grow quite large saplings in those. And this is just daily traces of transpiration here in pink. Through adding an instrument to this, like a psychrometer, we can see that the water potential declines as transpiration is going up, right? Until transpiration becomes limited, and then the water potential recovers. This red line I added is kind of interesting. This is that trigger loss point, right? Most plants do this. They're optimized to use water until they can't, right? Until they can't keep their stomata open anymore because they've kind of reached a level of xylem tension that will lead to trigger loss. And then overnight, they're kind of recovering as their transpiration is limited. These psychrometer instruments that measure water potential directly are really kind of finicky. They're difficult to install, and they're so expensive. We pay maybe for a logger in a chamber nearly $5,000 US. And we've got 28 of these in the lab, but it's not something we could scale. We could never put this out on hundreds or thousands of plants if we wanted to in like a proper screening system. But if you look at the organ diameter change, this is another feature that lately has come out of a lot of work from the Broadrib lab. We see that the branch diameter change, the branch shrinks according to transpiration. It tracks water potential pretty well. It's not perfect, The recovery of water potential estimated from this is worse than the decline in water potential. That's probably due to the lag of the volume refilling of the radial sap. But this is a pretty good proxy. So we're pushing forward with using it to predict water status in real time. I'll show you the instrument we 3D print and use for that soon. I just want to point out that it's really important to actually monitor the plant water status and not just think that soil moisture is going to tell you what you need to know. The soil moisture here for this plant is shown in the blue line. And you can see that soil moisture decline lags after the water potential and branch variation decline. That's because transpiration is really primarily supplied by water that's already in the plant. And as those kind of radial water stores are depleted, that soil moisture is able to come in as the transpiration becomes limited by turgor loss and start to refill things. But that signal kind of lags behind the physiological signal of the plant using its water to supply transpiration each day. This is one of those dendrometers doing kind of the branch variation. This is from a PhD student in the lab, Metal and Kant's dissertation work, just showing what it looks like on a single tree in a period of growth before an experiment began. At the onset of drought, those daily variations in stem width get really high because the plant can no longer draw on its stored water plus soil water. Now it has to draw mostly just on plant water, and so the stem gets quite a bit lower, and eventually the soil water is at such a low potential the plant can't take any more up, and so things really start to crash. But you can monitor recovery or not in plants, which Medellin did in this experiment. We deployed like 64 of these dendrometers that just take a picture of the stem and use changes in the area between pictures to calculate at a really high resolution those diameter changes. This can be done in the field. Instruments that are commercially available from like Tomsk are used to do the same kind of radial dendrometry work. This is from the master's thesis of Anderson Cinquini, another grad student in my lab. He's got a network of these things out in the field where he's trying to look in a silvicultural context. How do these like differences in maximum diurnal shrinkage or that stored water usage relate to provenances that have relatively faster or slower growth in the same environment? And so we've got a common garden where we have, provenances from over 300 wild populations representing the entire natural range of Pinus taeda. And he's instrumented up 16 of these clones that represent that variation. And they're each replicated. So he's got a total of 72 of these dendrometers. And he's like presently working this data up for his thesis, but we're really excited to take some of this more like high time series approach to understanding kind of determinants of growth. It was really fun to speak with Dave Drew, who had to leave before this seminar, about a lot of shared passions for that. And so now to kind of put all this together, that's just like what we can measure and how we might measure it if we want to do physiological phenomics. How do we put that together in a project? This is a project, a case study I want to share with you from our PinePhys grant. This is about predicting ideotypes, novel and extant from physiology, hyperspectral imaging, and SNP genotypes. So we've been looking at this common garden where each one of these gold points is a wild type provenance that was gathered, planted, and grown here in our garden in Gainesville, Florida. Before they were planted, we had them potted, hedged, and then we planted vegetative clones. And so there's replicated clones of every one of these provenances in a common garden growing here in Gainesville, which is kind of in North Florida. And so, yeah, this is really nice because we kind of have the species range-wide genetic variation growing in a single site where we can start to ask how much variation is there in these traits and what might be the more or less climate tolerant combination that already exists. This is already, I've shown you this map, the geographic distribution of where our plants came from. Here's where they came from in climate space. So over almost a meter precipitation gradient and an almost 10 C mean annual temperature gradient. So this is a really broadly distributed species that inhabits quite a wide range naturally of climates. When you're writing, you can't ever in the US get a grant unless you've already done the work. I know that's backwards, but trust me, it's what we have to do. And so we don't do all the work, we just do a little bit, a preliminary data collection, we call it. And so I went out with one of my postdocs and we sampled just 5 of these provenances I've shown in blue, and we measured some of these climate resilient growth and survival traits. A lot of these are the traits I just got done talking to you about. And we kind of manually labeled these values as blue if we thought this is the most resilient provenance that we found out of the 5, or red if we thought it was the most vulnerable. And what stood out to us at first was there's not a single clear one that's like, this one's all blue or this one's all red. It's all, the traits are kind of mishmashed, right? We don't have like a winning combination of all the traits in one tree. And so the impetus for this project and what we told the USDA is like, if you give us just $1 million, we'll go measure the rest of them. We'll see if we can do it. And then just for that alone, we'll head out to the forest and we'll see, is there a winner on the landscape already? You know, has evolution and time and variation in between populations, has it led to some combination that could provide a lot more survival time? And so, yeah, we also, in addition to traits I forgot to tell you about earlier, we measured the optimal temperature for photosynthesis. So we did temperature and gas exchange response curves to get the temperature at which carbon assimilation, stomatoconductance, and therefore water use efficiency are optimized in each one of these provenances. So that varies across provenances. We also did some early projections showing that under future climate scenarios, even the most thermal tolerant provenances are going to become growth limited earlier by temperature than they are today. So this is our first signal that there's a potential climate threat to growth, if it's not survival. So a postdoc on this project, another postdoc in the lab, Dr. Mantova, who we should all congratulate. She just got a permanent position at INRA. Yeah, so good job, Mary Lou. She went out and sampled 50 of these provenances. Under the first year of the project, we kind of subsampled the total set of provenances to get ones that represented all the geographic and climatic variation that exists across the range. So we think this is a pretty representative sample. And then we did the thing. You know, we did thermal limits. Now I'm showing you the cassette we made to hold pine needles instead of broadleaves, right? We did gas exchange. You know, these trees are 13 years old in the field, and so we had to get a 10-meter tall scissor lift and then use like a pole saw to get branches out of the sunlit canopy that we could work with. We measured the residual conductance and the xylem vulnerability. And when we do all this, what's really nice is we can start to understand what's the variation in these physiological traits across the range-wide genetic composition of the species. And all these trees growing in a single common garden climate, right? And we were, if I can just say, like, we were pretty floored. Rob is probably like, that's ridiculous, right? But like the conventional wisdom is that some of these traits should be a lot more constrained than others. And so these now have been standardized again to z-scores. So the units here are standard deviations from the mean. And what that means is for many of these traits, we get 2 standard deviations away from the average quite often in particular provenances. And so there are individuals that have really exceptional trait values for better and for worse. And when we sample at this kind of depth, you know, 50 different provenances with the full physiological phenomic workup, we can start to make predictions. And so to make those predictions, we use a mechanistic model called Sero. This was developed at INRA in France, where Mary Lou did her PhD. She's actually returning to that unit now for a permanent position after doing a postdoc on this project. Funnily enough, she didn't learn to use Sero when she was there. She was funded on this USDA grant to go back and collaborate again and learn how to run the model. And so she's taken these traits, and I'm going to show you just first four trees simulated. And what we can do in this model is we can use all of the traits, the xylem vulnerability, the residual conductance, the maximum stomatoconductance, and we can predict the survival time. How long until a tree stops growing, you know, it reaches its target loss point, and how long until a tree has hydraulic failure. It can no longer move water and will die. We have here for you the worst performer that we found is on the bottom, so it's going to die the first. The best performer is right here. The average is an in silico computation. We, you know, often in physiological studies, we go and sample 5 to 8 trees, and then we just average those values together, and that's what it is, right? And so this is our kind of representation of if you took all 50 and you just averaged them, this is what that would say. And then here's an ideotype. If we in the computer said, let's allow a tree to have the best of everything. Right? It may not be genetically possible. We're working on that this year because these are all SNP genotyped and we're doing genetic association testing to see which of these traits are linked or not. But yeah, if we play out the survival time animation, our worst one's already dead, you know, in a matter of, 30 days. Whereas our best survivor would live, more than 4 times that of the average trait value tree. And so there are already exceptional trees on the landscape that could survive much longer into a hot drought than the average or certainly the worst performer. I didn't point out the solid part here is growth time. And so this was a really exceptional tree in that it was able to maintain function for a really long time into drought, unlike a lot of other trees. It wasn't the only one that was exceptional though. It's at the top of the bar here. This is all 50 now, kind of showing the same data. And you can see that we had quite a few. There were about 11 trees that had really exceptional potential for survival time. And these aren't things we have to breed for because these genetics are just on the landscape today, right? Now, the work that remains to be done is seeing how to kind of combine some of these traits with the kind of growth potential that we need in a commercial setting. We can also take these models from Searle, and instead of just driving them with the standard climate like 30 or 40C, we can drive them with observed climate or with future climate projections. So these maps are showing you under a business as normal climate scenario. This is, by the way, the climate future we're presently on. The path we're on is a SSP2 4.5, if you're up on your CMIP language. Between now and 2099, this is the percentage of years that would be lethal for the worst performing province. Right here mapped in red being more percentage of years lethal. This middle panel is what's called fossil fuel development. This is sometimes called a worst case scenario or like a drill baby drill scenario where we start emitting more carbon. As an American, I was really happy to be busy all day yesterday, but when I got back to my room and I pulled up the news and I listened to the inaugural speech of our now president again, he said the phrase, "We are going to drill, baby, drill." And so maybe this middle panel, we shouldn't be like, that's ridiculous anymore. That's a little scary to think about, but whatever. When we look at the best provenance from a climate resilience standpoint, we see 0% of lethal years between now and the end of the century. So there is some real potential for resilience already in the existing genetic combinations on the landscape. Now, the ideotype, which is the fantasy case where we can have our growth and resilience together, comes out with the same type of solution. Finally, I just want to say, and I'm going to wrap it up in maybe the next 5 minutes, I promise, is that this all kind of assumes that water deficit is the major way to die, that, running out of water is what can kill a tree. But we really need to also be asking, and I hope I encouraged you to think about it at the beginning of the talk, is like how hot is too hot, right? Because even if you have water, if plant tissues get to a certain temperature, they can risk survival. So here's the 1990 to 2010 distribution of the hottest single day across the entire range of loblolly pine. Florida's here. That's the Mississippi River. It's too wet for loblolly pine there. It's actually really good for angiosperms that outcompete it. That's why there's a disjunct in the range. And so, it was never hotter than 44C, which is like our average, critical temperature for this species. However, if we look forward in the climate scenarios, the trees that got planted right before the COVID pandemic, that rotation will end and they'll be harvested by 2040. Some parts of the range are already going to be experiencing temperatures hot enough to kill, even before the current rotation ends. Right? What about the rotation after that, or the rotation after that, or the rotation after that? And so extreme heat stress, these kind of like touch it and die temperatures, are the kind of thing that we probably need to investigate a bit more. I already told you there's intraspecific variation in loblolly pine in these temperatures. So this is just showing the first 50 provenances that could vary, more than 6 degrees C. And so one of the things we need to know is this the kind of trait that can help us make predictions? The first thing to say about that is you need to know something about how plants respond when they encounter their thermal limits in dry air. These are thermocouples measuring the surface temperature of a Coffea arabica leaf. This is my favorite beverage. I had some this morning. And this blue line here is in the air. And we put these in the climate time machine and we take their temperature rapidly up past the critical temperature for this species at 44C. And you see that the leaf temperature, while it immediately follows the air temperature, the stomata open in a matter of moments. And effectively cool the leaf below that critical temperature. And this can be maintained for several hours, right? Until the little plant in a pot starts to run out of water. And so the thing I haven't told you is this coffee did this in the dark. So a plant will open its stomata in the dark and cool its surface to avoid these thermal limits of survival. And so when it happens in dry air, this is good to know. When we think about this in the What Kills Trees framework, this is a figure from Mary Lou's paper a couple years ago, showing that, originally a tree goes through building some xylem tension, maybe some of that xylem fails, there's cavitation or embolism, the water supply goes down, 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 until eventually you're past the point of no return, and now your membranes are damaged, maybe your meristems are damaged, and you cannot recover. The traditional view on how heat and heat waves affects this plant mortality mechanism is it's just a foot on the gas pedal, right? It just speeds, It's like one of my teenagers who just got her driver's license last year, my oldest. It just goes too fast. And yeah, certainly heat can accelerate dehydration and push a tree through this quickly. but also there might just be bypasses. You may not have to go through the entire spiral. We can just get straight to meristem damage if that temperature reaches a tissue that causes it to die, right? And so we need to probe these kinds of things. Furthermore, right now we just use like a heat intensity framework, like what's the temperature that could kill? And we need to start building response surfaces like this that integrate heat duration and heat intensity, while recognizing there's probably a temperature beyond which it doesn't matter if it's for a few minutes or a few hours. If you get that hot, you're gonna die. There are physical limits to living systems. So we put a bunch of pine trees back in this climate chamber system. I'm just showing you those same plots as before, and did a series of heat waves to see at what temperature do we go from plants that look like this to plants that look like this. The control condition had no heat wave. We were trying to replicate the 6 hottest hours of a day here in the climate time machine, so it went at 30 and stayed at 30. And then we did a bunch of temperature ramps going up to 60 C. This is actual data from the experiment in the boxes. There's 2 lines for each one because we put 5 plants in each 2 boxes. That was like a replicated design. And then to kind of compare this to the water bath treatment that's typically done to determine thermal tolerance, we built like the biggest thermal bath we've ever built. And of course, because money's not infinite, we used a trash can and a bunch of like sous vide heaters so that we could raise the water in the trash can from 30 C in those fine increments all the way up to 60 C. We had a big aquarium pond pump in the bottom to well mix this volume. It's maybe one of the more ridiculous things we've done recently in my lab. This project, by the way, was led by Emily, another PhD student in the group. And so we put 80 trees in that were well watered into these battle boxes. So they had lots of soil water. We took 80 trees and we put them in after a 2-day drought. So not enough to really cause any permanent damage, but enough so that they would hit their turgor loss point during the heat wave. And then finally, we dunk tanked 47 trees. All this material was clonal. It was generated by Arborgen using direct somatic embryogenesis. So we tried to have really genetically as homogeneous as we could plants for this study. Coming out of the battle box, you can see immediately, you don't need a physiologist to tell you that there are differences in the treatment on canopy survival, but we also do something a bit more quantitative. This is that chlorophyll fluorescence imager I mentioned earlier, and we took a picture of the upper third of the canopy of each of these trees. This is what those canopies look like across the temperature gradient. This vital signal is healthy if it's in this yellowish to green 0.8 to 0.7 range. And it's dead if it's down here at 0. And so as we go up to temperatures, you can start to see the temperatures that damage and eventually the temperatures at which things are just dead. This is the trees that went in irrigated. This is trees that went in after a 2-day drought and trees that went into the dunk tank. And already the kind of gold standard for thermal tolerance measurement, which is submerging plant tissues in a water bath, is overestimating the damage that we observe when we put whole plants through the same kind of experiment in dry air. So we might be in hot water. Is that an expression here? In America, that means we're in trouble. Yeah. Okay, and so this is just fitting that vital signal, the chlorophyll fluorescence response surface over temperature. And if the plants go in dry, the lethal temperatures may be 45.6. If they go in with soil moisture, not surprisingly, they're able to survive higher temperatures because they can use evapotranspiration to cool their surfaces. And then finally, the dunk taking a whole plant. And 51 C is what happens if we pull needles off the tree and put them in a water bath independently. So however you do it with hot water, we're maybe overestimating how much heat plants can take. We do think that the mechanisms of death here are not hydraulic. The wet trees maintained a really hydrated water potential, while the droughted trees, as expected, used more and more water. They basically went to their turgor loss point, which is about 2.1 to 2.2 megapascals in this species, and then stopped losing water because pine has a really low residual conductance. None of this is low enough to cause embolism or hydraulic failure, which is that major mechanism of tree mortality. Yeah, I guess just last thing to say is we're monitoring the plants now. We want to see what's happening with meristems. We're getting epicormic resprouting in the trees, and probably we expect that the meristems are more thermal tolerant than the foliage of the trees. So this is kind of interesting to see. But those meristems also seem to have a different sensitivity based off of whether or not the tree was droughted. So Emily's still observing these plants for another month or so, and then we'll submit the paper. I showed you this map for loblolly pine. Last night I stayed up to make one for South Africa. This is like single hottest day, and I like tried to overestimate the temperature by a degree because I've not measured the pines in South Africa. Let's say it was 45 instead of 44. The red areas are where you're going to have a single hottest day that exceeds that lethal temperature. I did a version of these too. The areas that are getting over that temperature are getting over that temperature much more than one day. I think the single location that went over 45C the most did it like 83 days of the year or something under 2060 to 2100 climatology physiology under a worst case scenario. So it is possible that even here in South Africa, we're really going to need to be thinking about moving from these safe observed climates to these potentially risky climates that, the mechanism of mortality might differ. It might be based directly more on heat damage than hydraulic damage. Certainly, they're going to interact a lot. If you like anything I've said, it was probably these wonderful people. The lab that I have back in Florida is full of really talented and brilliant graduate students, undergraduate students, and postdocs. They are the backbone of the work that we do, and I'd be happy to take any questions.
Well, thank you very much for a fantastic seminar. I think the feedback that we heard on the mic is the system going up.
Yeah.
Some really concerning, some really concerning information that you're sharing. I mean, I think we're all aware of the threats that are there, but the way that you're quantifying that is really illuminating and really digging into these mechanisms, really, really important. So thanks a lot for that fantastic seminar. Let's open the floor for questions or and one of you just keep an eye for me online. Those of you online, just put up a hand and we'll unmute you. All right, questions, Rob.
Thanks, Bill. As always, you generate these thought-provoking talks, so that was fantastic. Thanks. I have a few questions, so I'm going to try and pick out the one that I think is the.
I promise to go to dinner with you too, so you pick your one or two. Yeah,
great. Your study with Marilu, you have your different genotypes that are responding very differently in terms of their time scale. Have you analyzed what's the mechanism behind those differences? What are the key traits?
Yeah, so we have done a sensitivity analysis. This is in the paper that hopefully I just sent the final drafts comments back to Mary Lou like the night before last. And so hopefully that paper will be submitted soon. But the key traits, the residual conductance was a major one, because when you think about how our current mechanistic models are built, right, that's the water loss rate, the leaf area index difference between these trees was different. So kind of canopy architecture matters a lot. We've currently estimated this canopy architecture with allometric equations, but another PhD student who works in a colleague of mine's lab at UF has just done TLS LiDAR scanning of all 50 of these trees so that we can get a more accurate estimation of that. But yeah, those are the two that were the most important. Xylem vulnerability was also important, as was the Turgor loss point. There were quite a few traits that were we were kind of surprised, like capacitance didn't show up in that analysis. But when you look at the contribution of these individual traits to the model performance, G-res, that residual conductance, like by far and away, if you could only measure one thing, that was the one that seemed to matter the most.
Yeah,
at both simulations of 30C and 40C, and we measured it at both controlled temperatures too. So we actually parameterized the model with different trait values based on the real observation.
Okay, fascinating. So just an observation on the growth of the individual that lasted the longest, that was beyond the point at which the other genotypes had already died, right?
Mm-hmm.
So and that would be linked to, so that's not linked to the G-res, that's linked to the canopy architecture.
The canopy architecture, yeah, also probably a lower GS max, if I'm guessing. I would need to look again to be sure, but yeah.
Yeah, these mechanistic models seem to be the best way to try and predict future plant responses. We've come from this kind of simplistic trait frameworks, and really the mechanistic models do seem to be the way forward for doing so.
They do, and when you look at the individual trees, there was like a little less than a dozen trees that had exceptional survival time. They don't all do it the same way. Those aren't like the 11 best G-res values. Some of them, that's how they're doing it, but other ones are doing it with different trait combinations. And so this is probably a case in which we're going to start getting into thinking about the concept of trait equifinality, or basically getting the end same functional result through multiple trait means. And so just like there's many ways to survive, there's probably many ways to die or to avoid it. And I totally agree with you. These models are key. At the moment, they're a bit incomplete. For example, there's no mechanism for heat-induced damages or death in Siro, which is probably the most advanced plant hydraulic model. But that's something Mary Lou and others are working on now. Yeah. Mhm.
Other questions?
Great talk. It might be a stupid question, but I just wanted to ask, like, when you did your sample collection in field, right?
Mhm.
You guys were talking about the canopy where you are literally taking the pine. Is there a specific height that you focus on, and is it a constant height? Let's say you say 100 meter or something. Is it a constant height that you go for all the plantations that you look at? And after you collect your sample and go for testing, how many hours can you say, okay, it was not dried up based on traveling and stuff like that?
This is actually, I just want to point out, this is a great question to answer your first part of your question. Yeah. How we, especially when we're doing physiological phenotyping, how we treat and handle our samples, where we sample our from, and how consistently we are in sampling is super important, because the variation across a canopy can be as big as between two individuals. And so, yeah, we typically sampled the upper third of the sunlit canopy on the south facing side, because this was done in the northern hemisphere. So we wanted the sunlit upper third of the canopy. Regarding the height varied quite a bit, because you remember this garden has the genetics that represent the species range-wide genetic diversity from wild type collections. So we had some trees at 13 years that were 8 meters tall and some that were 15. And so we decided developmentally to stay in the most, the upper third of the canopy. The samples, the measurements that were done on a tissue basis were typically done on the most recently fully expanded, matured foliage, the fascicles that were most recently fully matured and expanded, and traits that were conducted on woody tissues were taken at like the internode between those needles and the prior year's growth. Yeah. So we tried to keep it all developmentally constant. Yeah.
So you're talking about like there are other trees that are very tall and some that are short.
Mhm.
Did you see like a difference between like the impact on the shorter ones because maybe the sun is not hitting too much in comparison to the ones that are taller?
Yeah, and so something I didn't go into detail because I was probably going too fast already is in this garden, we have 6 replicated blocks. And so we can measure the individual in 6 different conditions. So we're hoping that kind of accounts for the neighbors are randomized within those blocks. Some of that variation that may be caused by one neighbor or another. But yes, in the end, there are definitely effects of the fact that these trees aren't all being grown in like, genotypically homogeneous blocks. Because there are differences in height. And so that's going to change things like the leaf area index. But by sampling those replicates across the garden where those other variables are changed, we're hoping that maybe we've accounted for that a little bit. I also realized I didn't talk about how we made sure samples didn't dry out. We collected things in the field early in the morning, which made it more difficult because we are gluttons for punishment. We then put them in like big trash bags with, humidified towels to make sure that we really limited the chance at transpiration. We then typically chilled the samples, not at a freezing temperature, but at a refrigerated temperature to slow metabolism until we're ready to work with them. We typically took all our physiological traits within 48 hours. Some sampling we did at the beginning to understand this. We think that with these excised branches like this, we can work with them on most traits for up to about a week. Gas exchange traits can be a little more sensitive than that. Some of the traits like xylem vulnerability seem to work in pines at least for up to a couple of weeks, so long as the samples are well stored and well handled. Yeah. Mm-hmm.
Yeah, Bill, sorry that I'm in the back, but I enjoyed your seminar nonetheless.
Good to see you.
I just wanted to ask you, your system actually lends itself very well to looking at how oleoresin would affect the whole resilience to drought and to heat stress. So have you ever already thought about doing these experiments, or is this something that you will attempt to do in the future?
I have thought about it. It's not getting at like the oleoresin, like chemistry or composition, but a trait that's kind of maybe tied to this a bit. The master student I showed, Andrew, that's doing the dendrometers, he's also going back out and doing tree coring now that we've collected all the physiological traits, and is going to be doing some resin canal anatomy, looking at resin canal density and size variation between these. We might use that as a first attempt before we try and find a partner on chemistry. But no, it's not something we currently do. It would be really cool because this is a really important part of the defense system of a pine tree against biotic agents. Yeah.
Chris, and then I come to you, Zamishka.
Okay, I'm going to ask the token entomology question.
Yeah.
I'm an ecophysiologist as well, but I work on insects, with working on water balance and thermal tolerance. So with the models that you've been looking at, it's a big stretch, but what kinds of implications can you draw for how the plant going through water stress, like you're modeling, is going to affect the way that the insects feeding on them might be affected?
Yeah.
For aphids, for example, or other sap-sucking insects, which in the literature for insects, because they are capable of losing water through that and able to cool themselves evaporatively, if you remove that source of food, how is that going to affect them?
This is a really good question. I don't know that I'm well qualified to answer it because I don't research this. I will say that generally the approach that we're taking when we look at differences in growth suppression, when we look at like that turgor loss point I was talking about, we do know that the metabolism of a pine tree slows kind of significantly after this point. And so the generation of additional defensive compounds is probably going to be slowed relative to when the tree's in a more vigorous hydraulic position, when it has more water. And so I suspect that this might have a kind of a positive effect on biotic agents that might be wanting to attack the tree, but I wouldn't know for sure. Yeah.
Thanks, Paul.
Mhm.
The question I have is related to what helps trees survive. And one of the things is the contribution of the roots themselves. And how are we modeling this and incorporating that in our prediction models?
Yeah, this is a really good question. So this Searle model allows you to have kind of multiple rooting zones. You can parameterize the root hydraulic traits if you have measured them. I want to be really clear, we did not. We made some assumptions about more even root hydraulic characteristics. It's probably pretty obvious, but doing root hydraulics is really difficult because one, you got to have a strong back and a sharp shovel, and two, you got to have like some sort of resilience that even the trees don't. But it is a really, really important question. So mechanistically, these models have the ability to parameterize a lot of those root traits, which is nice. On the flip side, we don't often go out and measure them. However, these models, as kind of like a simplified representation of the system, can be a really cool way to probe that question, because we can take the trait data that we did measure above ground and then alter what's happening below ground. Well, we can kind of wiggle those parameters around to see if observations that we make about tree mortality in landscapes maybe are being maybe our model's plus or minus wrong due to something different underground. And so through these kind of like different iterative simulations, we can maybe reveal, if we are going to go underground, what's the best way to do it. In my lab, we've been in other systems working on developing the ability to do hyperspectral imaging below ground. We had a paper out last year on called Signals in the Soil, where we used a hyperspectral imager to independently measure the root water content and the soil water content. We think approaches like this through time might be nice, because the long-held assumption that the roots in the soil are in some sort of hydraulic continuum only holds over a certain range of conditions. And we don't know too much about what happens once plants fall into this like hydraulic disequilibrium. So we've got some interest in a few directions we might push into to get into that. The models are ready to receive the traits, but if you go out and try and collect global trait data on below ground, conductances and resistances and things like Rob, there's like a handful of them, right? And so at the moment we're kind of at that remains a black box, and that's a problem. That's a problem. The systems in which it's tractable are really small plants, but you'd like to know it for big trees like these. Yeah.
All right, I'm going to just double check if there's anybody online with a question or a last burning question in the room. Online, I don't see anything online now. Perhaps a very last question for me. I mean, you referred to upscaling some of these, our ability to measure some of these traits so that it can be incorporated potentially into breeding programs, for example. Through your work with Camcor, with others in the US as well, to what extent are companies starting to look at integrating these traits into breeding programs?
I think they're serious about it. There are several companies that are breeding eucalyptus that have reached out afterwards. One of them talking with kind of intensively about developing an in-house physiological phenomics platform. And so I think there's starting to be this realization that focusing on yield and MAI and these types of traits only is great, so long as the going is good. But as we have more and more problems cropping up, whether you want to call it physiological disorder or other things, there's a need to start understanding and screening for traits that need to be kind of like bred into or brought into those breeding populations and into the trait distributions that are being worked with, so that the coming climates don't become problematic, right? Because when you're working in long-lived perennial plants that have a rotation of 8 to 20 years, the plants that did really well when you screen them today, by the year 2045, they may not be doing so well. But we really need them to survive until then if we're going to harvest pine for timber, for example. Yeah.
Right. Yeah, I think, well, it's very it would be obvious to everybody in the room, and I'm sure for those online as well, why we're so excited about working with yourself and the interactions we had this morning with Dave and people like Rob and others that are working in this field. To layer on top of this the biotic component, which I think that's a black box that we also understand very, very little about, but is absolutely crucial to also get into these time machines of yours.
You need to load them up with biotic agents. Yes,
absolutely. Paul would know that I'm really, really keen to have some of these time machines here and start addressing some of these questions. And I think those are the things that we want to be doing together, taking the strength that is here and understanding biological systems and the understanding of what's happening in terms of climate and tree physiology, and starting to layer these things on top of each other, because certainly is a multi-layered question that we need to understand for the future. So we're excited. We thank you again for a fantastic talk, and we're excited about continuing along this road with you. Let's give him a last.
Thanks for the invitation. I just want to say thanks to everyone for coming. Really, really lovely questions, and it's been